Okay, today we're going to talk about RNA splicing in general, partly as a review, but the main purpose here is to examine some recent therapeutic approaches where RNA splicing is modulated in order to treat genetic diseases, application of the basic biology to something that's therapeutically extremely important. Okay, so in general, splicing, we've seen the GU-AG rule. Usually there's GU at the 5' border of the intron, within the intron, AG at the 3' border, and most of our introns have these sequences. The other crucial part of the intron is an A, which is next to a polypyrimidine track near the 3' border of the intron. This is where the lariat structure will form during splicing. And there are other sequences, including within and adjacent to the intron, that will facilitate uses of these 5' and 3' splice sites, etc. Okay, so remember SNRPs. Splicing is accomplished by these small particles that are made of a uRNA, small RNA, and the associated proteins. So these are called SNRPs. So the uRNAs are U1, U2, U4, U5, and U6. I don't know what happened to U3. Anyway, each has its own associated proteins, and these are the ones that carry out the splicing reaction. Okay, so this is a rather detailed picture of the process. You certainly don't need to know this, but it's important to envision what's happening during the splicing reaction. First, we have our pre-mRNA, which contains an intron, and we want to remove it. So these 5' and 3' splice sites flank the intron within it, and then we can see the A, which is going to be the location where the lariat's formed. Then we have a very orderly series of events where the uRNA SNRPs plus other splicing factors associate with the intron and eventually accomplish the splicing reaction. Okay, so this is an orderly series of events where these various protein RNA complexes will join and eventually carry out the splicing reaction. Okay, there are, besides the uRNA SNRPs, we have splicing factors, which in this picture are going to facilitate the splicing process, facilitating the association of the SNRPs with the intron in the proper order. Okay, so they don't act alone. They've got other proteins that are involved. One is the U2AF or U2 associated factor proteins, which facilitate the process. There's also the SR proteins, serine arginine proteins, named after their abundance of those amino acids. These are also positive regulators of splicing. But in the end, we remove the intron in the form of a lariat, in which the particular A at the branch point is joined using its 2' OH to the first nucleotide of the intron. So the process finishes when the exons are ligated together. And of course, most RNAs have multiple introns, so this process would occur repeatedly during the course of preparing a pre-mRNA for eventual translation. In terms of cis sites and trans factors, there's a lot more to it than just the GU-AG rule and the branch point. Okay, we will have a series of RNA sequences which can bind the splicing enhancers or the splicing silencers to determine whether or not a particular intron is used within a particular mature RNA finally. Okay, so what that means is, for example, we know that alternative splicing is used quite widely in mammals, where we would choose which exons to include and which to exclude for the final product. Okay, so that will give us different protein isoforms many times. Okay, but we've got to have positively acting sequences as also negatively acting ones in order to choose which exons to include or not include in any particular final mRNA that's formed. Okay, so some of these negatively acting proteins include the HNRMP proteins, which negatively regulate splicing. So proteins of this general type can cause exons to be skipped. And again, we're going to have proteins that recognize specific RNA sequences, either in a positive or a negative way, depending on what their particular role is in that process. Okay, some of the sequences that might be present in various mRNAs, pre-mRNAs, are exonic splicing enhancers, which are what they sound like. They're sequences which bind RNA facilitating proteins that occur within the exon. So we could have splicing enhancer proteins which bind within the exon itself. Also, we can have exonic splicing silencers, which is what that sounds like also. Within the exon, we have a sequence in the RNA which binds a protein which silences or prevents use of that particular exon in splicing. Then some of these sequences are located within introns. ISE is intronic splicing enhancer or intronic splicing silencer. So in essence, these sequences bind proteins which either enhance or reduce splicing efficiency. And in general, this is accomplished by interacting with the SNRPs, which are, of course, the core RMPs involved in the splicing process. Okay, introduction to our genetic disease, where understanding splicing has given rise to excellent and now FDA-approved therapies for this disease. Okay, SMA is spinal motor atrophy, and it results from a defect in a gene called SMN1, which is survival of motor neurons. Okay, most of the disease cases are actually deletions of the SMN1 gene, either the whole thing or big chunks of it. So people who are homozygous for these deletions have no good copy of the gene. They have this disease, spinal muscular atrophy. Okay, and the disease is recessive, but it's fairly common, really. 1 in 50 people are carriers. The disease itself involves motor neurons that start degenerating very early in life, probably before birth. And there are various forms of this disease which depend on how extreme they are, but the severe forms are almost invariably fatal. Now, the protein itself is actually distributed through the body, but the effects on the nervous system are what cause the fatal effects. Okay, around 1 in 10,000 newborns afflicted, which sounds rare, but it's not really. That's a lot of people. The disease is not yet part of newborn screening programs yet, but it will be, because there was no therapeutic intervention possible, but now there is. So that is set to change, I do believe. Okay, so the hero to the rescue here is SMN2. SMN2 is a paralog of SMN1. Okay, so what that is, it's another copy of the SMN1 gene, essentially, that arose from a gene duplication early in human history, in our case. So that describes a paralog, that the genes are related by a gene duplication. So SMN2, we each have from each of our parents one copy. So we have each of us one from mom and one from dad. Now, importantly here, which gave rise to the science that led to this treatment, is that some people have extra copies of SMN2, which is a result of an additional gene duplication or some other sort of rearrangement. So SMN2 is like a backup copy for SMN1. The problem with it is it's only partially functional. It's a weak version of SMN1, and even though it's actually transcribed and translated, it does not provide adequate amounts of protein to effectively treat people or to make up for the lack of SMN1 in the people. And that's a key observation. Okay, so SMN2 is a weaker version of SMN1, because even though you do produce protein from it at some level, the mRNA gets degraded because of exon skipping in SMN2, which we'll get to in a second. So a little bit of SMN protein in SMA people comes from their SMN2. We do have some copies of it, and some protein is produced, but not enough to make up for the absence of SMN1 because of this exon skipping problem. Okay, so diagramming this, unaffected people, of course, we express SMN1, we get functional protein, no problem. We express SMN2, but it's mostly non-functional, which is the normal situation in most people. And we're showing only one copy of each, but of course we have two because we have two parents. In people with SMA disease, the SMN1 is inactive by deletion, and the SMN2 is producing protein, but not nearly enough to make up for the absence of SMN1. Okay, so in terms of types, type 1 is a severe form, and children with this form of disease rarely live to be older than 2 or 3. They never are able to have head control, turn over, sit up, and they ultimately have serious breathing difficulties because the muscle weakness is progressive. Types 2 and 3 are less severe, but still very debilitating. Type 3 is the milder form, but it still involves progressive muscle weakness and later inability to walk or climb stairs. Okay, so the important observation here is that in looking at the genotypes of the people, the severity of SMA is related to the SMN copy number. Now, they're dividing the types of disease into type 1, type 2, and type 3, where the non-sitters are the ones in type 1 with the most severe form. And most of them have the expected number of SMN2 genes, which is 2, one from mom and one from dad. But there are occasional people which have an extra copy. In the next version, type 2, where kids are actually able to sit, at least part of their lives, they actually tend to have an extra copy of SMN2. So they have 3 copies instead of 2, and that extra copy is enough to make their disease less severe. Moving on to the less severe types yet, those people have 3 or 4 copies of SMN2. Again, to the least severe effect, people have 4 copies of SMN2. So this gives us the idea that SMN2 may be non-functional, but it's not completely non-functional, and that providing extra SMN2 protein would be a treatment for SMN1 deficiency. And that's the basis of these treatments that have been approved recently. Okay, so in summary of that, the severity of the SMA that a child has is proportional to the number of SMN2 genes that they have. That's because SMN2 is the only active source, but extra copies mean more active protein and less severity. So SMN2 is not totally non-functional, but it's not functional enough to make up for the deletion in the SMN1 protein. Okay, so this just shows the types: infantile onset, 2 copies; later onset, 3 or more; later still onset, most have 3 or 4 or more. And some people can walk, but later end up in wheelchairs. So there is a great diversity in severity, which is largely attributed to the extra versions of SMN2 that the people have. Okay, so the idea is if there's more active protein, progression is slower. And in these patients, clearly extra SMN2 can substitute for the SMN1 deficiency, at least to a certain extent. Okay, so the question here is, why is SMN2 so weak? The protein coding region of the gene is actually exactly the same as SMN1 if you just look at the DNA sequence. But it turns out that exon 7 is often skipped during splicing, with the result that those amino acids are not present in the protein like they should be. And as a result, the protein and the RNA are both unstable and can't function. So the idea here is to try to improve the inclusion of exon 7, trying to kind of fix up the SMN2 gene so it's not so weak anymore, so that more of the protein could be made. And that is what was done. But this depends on a careful analysis and understanding of the splicing process in order to figure out how to fix it to make exon 7 become included. Okay, so here's our picture again. SMN1 affected and unaffected people. We mostly produce non-functional SMN protein from SMN2, and the reason for that is we're skipping an exon during splicing. So we need to look carefully at the splicing of that exon to try to figure out what's gone wrong in order to try to fix it. Okay, so why has it gone wrong? It turns out there's only one relevant nucleotide difference between SMN1 and SMN2, and that is the substitution of a U for a C in the SMN2 gene relative to SMN1. So in SMN1, because of the C, exon 7 is included during splicing, and you get a complete protein which is functional. In SMN2, with the mutation, there is a skipping of exon 7, with the result, most of the time, with the result that you mostly get non-functional protein out of it, unstable protein and unstable RNA, but you do get some. Okay, so what this is believed to have happened is that we took that U mutation has contributed to an actually intronic splicing suppressor, or negative silencer rather, and that's responsible for the skipping of that exon. So that U is basically having interactions with an adjacent intron, which is ultimately causing that exon to be skipped. So what happens is that nucleotide interacts with proteins that are bound to the adjacent intron, forming an intronic splicing silencer. So in terms of approaches, we're going to try to figure out how to improve that based on our knowledge of the situation. Okay, there's another approach which I will mention later where there's actual gene therapy involved, where we're giving the people actual SMN1 protein, gene back again, and that's also working. Anyway, we're talking about splicing now. Okay, so diagrammatically, the SMN2 targeted therapy, if we have no treatment, we get a little bit of SMN protein from SMN2. The ideas are we're going to use an allele-specific oligonucleotide, which is targeted at that intronic splicing silencer to try to hide it from the splicing machinery so that we can get correct splicing. And that's the one we're going to talk about most. The other approach was actually developed by a drug company which actually identified small molecules that facilitate U1 interaction with that intron, and that is another successful approach. The third successful approach we'll talk about very briefly is an actual gene therapy where the person is actually given an active form of the SMN1 gene, which hopefully is believed that it will keep them fine throughout life as opposed to the repeated treatments. So we'll see how that works out. Okay, so the oligo masking idea here. This oligo has two different names. One is nusinersen and the other is Spinraza. It's marketed as Spinraza. I don't know why they change the names of these things all the time, but they do. Okay, but the idea of this masking is that we're going to give the cells an oligonucleotide. This is a DNA oligo. And what it's going to do is bind to the sequences within the intron that are recruiting the silencer proteins, thereby competing with them. So the idea is that without this oligonucleotide, we have the HNRMP proteins binding to this intron rather, and causing the adjacent exon to be skipped. It's because of the binding of those proteins. So the idea is we're going to put a oligonucleotide which binds to the RNA at the site where the protein would otherwise bind, competing with it, and therefore preventing the skipping that would occur as a result of that silencer. So essentially we're going to inactivate the silencer by not allowing those HNRMP proteins to bind there. Okay, much work later. Here is going to be some experiments that identify the oligonucleotide that's going to perform this function for us, which is to say identify the sequences that are needed for correct splicing and identifying an oligonucleotide which can prevent the activity of the silencer, therefore allowing us to treat the people with the desired oligonucleotide. Okay, so what this describes is what's called a plasmid minigene experiment, where you have the exons that you're interested in with blanking introns and placing them into cells, and then asking whether the exon is or is not included, depending on what mutations you have in it or not. So we're going to be basically looking at the products of splicing in a series of plasmids that contain this particular exon in the presence and absence of oligonucleotides later. All right, so we put part of the DNA encoded in the introns and exons in a plasmid that has a promoter for expression in mammalian cells. That's important. Then we're going to isolate RNA from the cells, reverse transcribe it, amplify it, and then analyze it. In this case, we're going to be looking at the sizes of the PCR products in order to tell us what actual exons are included and which ones are excluded. Okay, so the first thing is to identify this splicing silencer by making deletions. Okay, so the idea here of making deletions is if you have a sequence which is causing the effect of silencing, then if you delete that sequence, you will find that the silencing is diminished. So we're basically identifying the sequences that are doing the work to prevent the splicing And by removing those, or by identifying what's removed that's important, we'll be able to define the silencer. Okay, a lot of them look pretty good, but oligo 10 to 24, which is in lane 13, has only 3% skipping, which is basically as good as the larger deletion. So that would be the one we would think about making or using as identifying the target. Okay, so that silencer is really not, you can be inactivated by removing a fairly small number of nucleotides from the border of that intron. So that basically defines the silencer as something that occurs between nucleotides 10 and 20 in that intron. So that's what we're going to want to try to target with our allele-specific oligonucleotide. This deletes the site, but that tells us what we're looking to kill, which we can do by binding a complementary oligonucleotide to it. So this takes a little bit of careful looking and thinking to kind of get straight. Okay, so this basically reiterates what I just said. You've got the piece of SMN2 that you want to examine cloned into a plasmid and put into cells. Each row is a plasmid that has a part of the intron deleted. In general, the word delta means deleted. Okay, and each row has different nucleotides, and that you can look at the effects of those deletions by looking at the gel underneath. So the gel is correlated to the roles in the diagram above. So in each lane, we can tell which band is which. The upper band on the gel has exon 7, which is to say the lower one does not include it. So the difference in size is because of skipping. So we just go through the different deletions one at a time and see which ones give the most improved inclusion of exon 7, and those define the important sequences in the primer. So it looks like nucleotide 10 to 24 is as good as any, and so that's what we're going to say is the important part of our silencer. Okay, so the reverse of this is what we really want to do, which is to say to mask the silencer in people. So we have to design allele-specific oligonucleotides that can bind to the silencer and therefore reverse its effects. So this is kind of the reverse experiment of the previous one, because then we were looking at the effects of deletions. So now we're going to look at the undeleted one to see what we can bind to it in order to prevent HNRMP binding and silencing. So that should be ideas, of course, if the oligonucleotide can compete with the HNRMP proteins, that exon 7 will be included and the full-length protein will be made. So that's what the next experiment is that we're going to look at. Okay, so this is a similar splicing assay, but kind of in reverse. Well, we're going to look to see whether if we add antisense oligonucleotides that will bind to our intron, either intron actually, to see whether we can mask the activity of the silencer. So what we're looking for is the inclusion of exon 7 in a way that depends on the presence of a specific nucleotide that is expected to bind to that adjacent intron. So again, we've got the same controls. SMN1 is on the left. Okay, we can see that if we look at the splicing assay of the SMN1 gene, that the upper band includes all 3 exons, in which exon 7 is included. The lower band is so faint you can barely make it out, but that would be exons 6 and 8, which would be those that actually excluded the exon. Okay, the next lane is essentially SMN2 without any oligo, so that's our starting point for this. And you can see that most of the RNA excludes exon 7 because mostly it's the lower band. There's a certain amount of the upper band, but not nearly as much as SMN1. So the rest of the lanes have different oligonucleotides added to the cells and asking which ones give the best retention or inclusion of exon 7. So what we're looking for is an intensity of the upper band indicating that exon 7 was included. And so you can march along your intron and say very clearly, looking at it, the oligo labeled 11-25 had almost complete inclusion of exon 7 versus many of the others. All the others basically have varying amounts of exon 7 inclusion, but definitely 10 to 25, it does the best job. There's also some funny business going on in intron 6, actually, where there seems like there's a negative element over to the left. But that doesn't concern us now. What concerns us is that nucleotide 11 to 25 very efficiently improves this correct splicing of exon 7 in these cell lines. So that gave the best one, almost complete, 96%. So that's the one we're going to go with, which matches the results from the deletion experiment, of course, pretty well. Okay, so in summary of these experiments, we have defined an intronic splicing silencer by deletions, where if we get rid of the silencer, if we get rid of that sequence, then splicing will occur properly. And if we have an oligonucleotide that binds to that sequence, hiding it from the splicing machinery, that has the same effect. It allows the exon to be included, which is what we want to do therapeutically. So our antisense masking model is going to be just that. We're going to use that antisense oligos, not just in cell lines, but also later in animals, and then as treatment for kids. And this does seem to work in this setting. So the oligonucleotide is now called nusinersen. I don't know why they changed the names of these, but they do. And so summarizing the mechanism of action is we have the SMN2 gene, we have the SMN2 pre-mRNA, and we can see in red, exon 7 is the one that would normally be excluding. Our antisense oligonucleotide binds to that, and therefore allows it to be included in the final mRNA. So the presence of that oligo allows proper inclusion, failure to bind the H and R and P proteins, and production of the full-length SMN2 protein, which is what we want from all of this. Okay, so this went through clinical trials, and it's now very widely used. It's injected directly into the cerebrospinal fluid, and it's given at 2-week intervals at first, and then every 4 months after that. And there are children now that have been on this for a couple of years, and they're mostly doing very well. The oligo is sold under the name of Spinraza. Okay, as far as early studies to show that this is true, when they do clinical trials, usually they have a placebo population, but it was considered that would be just terrible to have, kids that you got this drug and they get the placebo and so they die. I mean, that's terrible. So what they do instead is look at historical data, children with these disease and how they progressed, rather than having a control group within the clinical trial, which is really the only ethical way to carry out such thing. But the idea is that kids with a severe form of the disease will eventually have respiratory problems, that they have to be on respirators all the time. And so their assessment of success, in this trial at least, was ventilation-free survival, that they didn't need respiratory support for that amount of time. And you can see that in the nusinersen study, the kids did very well, and most of them actually never needed a ventilator. And this has also been used on older patients and those with milder disease, and it turns out that it improves their condition. Rather than stemming a decline, it actually improves their ability to walk and remain free of having to use a wheelchair, etc. So this has been a very successful drug and continues to be. So I said this before, there were no children given the placebo, fair enough, and the treatment greatly slowed or halted the progression of the disease. In another approach, and I'm not going to explain this too carefully, Roche Drug Company, well, all drug companies that do this sort of thing, have enormous chemical libraries, and so what they did was screen for molecules, small molecules, that would, by a similar assay, improve correct splicing of SMN2. And they found some, and those are actually now marketed because they were successful, and those can actually be taken orally. So there's another approach besides the antisense oligo, a small chemical compound, which is basically targeted at the same part of the process. And finally, there's an actual gene therapy approach, which is almost miraculous. Okay, I'm not going to go through the biology of the virus because it's a very complicated system. But what it is, it's what's called an adeno-associated virus. It's a very popular one for gene therapy, and that was used to basically clone and express the SMN1 gene in order to just basically replace the defective gene in the kids. So briefly, AAV is an adeno-associated virus. And what's done is that the DNA that you want to introduce into the person is encapsulated into the virus. Now, what's given to the people does not have any viral DNA in it. It's set up so that you get cell lines which will actually package the DNA of interest into the virus without actually including any virus itself. And that's a complicated process. Anyway, so envisioning this, what you have is your transgene, and that's what's carrying SMN1 gene under the control of a CMV promoter that is put into cells in such a way that all the packaging or all the viral proteins are being produced by the cell line, and that will package your desired transgene into viral particles that can then be harvested from the cells. And that's what's given to the people. Okay, we don't need to know this part. Now, the bad news of all this, these are 3 effective treatments, is the cost. The gene therapy one is the most expensive one. It's the most expensive medicine in the world. It's given only once by intravenous infusion, and it's given to children that are younger than 2 years, and it's $2 million, which is an awful lot of money. On the other hand, it's supposed to be a lifetime, essentially a cure that goes through the person's body and provides SMN protein for whenever it's needed, wherever it's needed. The oral type, which is a small molecule type, is less expensive and more convenient, and may actually be the drug of choice after a while. The Spinraza is the intrathecal injection, which is into the cerebral spinal fluid, and that's not exactly cheap either. So none of these are affordable, really, but insurance companies and drug suppliers have been very good at working with people to make sure that the people that need these drugs actually get them.